main immunoglobulin in the secondary immune response. The second antibody is the IgA, which is either present as a monomer, but IgA found in the secretion is a dimer, which is consists from the two antibodies, so it is called dimer, and the molecular weight varied between 150,000 to 600,000 Dalton, because this is, if it is monomer, its molecular weight 150,000, while when it is dimer, so it is 600,000 Dalton. When IgA exists as a dimer, a J chain is associated with it. This is the J chain that holds the two antibodies and prevent them from dissociation. When IgA is found in secretion, is also has another protein associated with it, which is called the secretory piece. And this secretory piece is made in the epithelial cells and is added to the IgA as it passes into the secretions. J chain linked to the carboxy terminal portions of the heavy chain, so the carboxy end of the two antibodies. Properties: IgA is the second most common in the serum immunoglobulin and constitute 10 to 15% of the serum immunoglobulin. IgA is the major class of immunoglobulin secretions, tears, saliva, colostrum, which is secreted from the breast of the mother after delivery, which is important in the protection of the baby from the disease. So we encourage the breastfeeding due to presence of the secretory IgA in the colostrum. Mucus also is present in mucus. Since it's found in the secretion, secretory IgA is important in local mucosal immunity. IgA does not fixed complement. IgA can bind to some cells, polymorphonuclear cells like neutrophil and NKT cells, and mediate the ADCC, antibody-dependent cell-mediated cytotoxicity. J chain is a small glycoprotein that are covalently linked to the carboxy terminal portions of the heavy chain. While secretory component, it is a polypeptide chain synthesized by the exocrine epithelial cells that enables IgA to pass through mucosal tissues into secretion and protect the IgA from protease enzyme that presents in the intestine, because the IgA is a protein and degraded by the protease enzyme. So this secretory piece, which is protect the IgA from degradation by the enzyme. IgM, the structure is exists as pentamer, which is consist from 5 antibodies: 1, 2, 3, 4, 5. So it's heavy antibody. Its molecular weight is about 200,000 Dalton. In the pentameric form, all heavy chains are identical and all light chains are identical. Thus, the valence is theoretically 10 times more than V1 antibody. IgM did not have a hinge region. As we see, there is no hinge region in these antibodies, and replaced by extra domain on the mu chain CH4, because the standard antibody has 3 constant domain, while the IgM has an extra constant domain CH4, which is replaced the hinge region. So it is so it has 4 constant heavy domains, and it has another protein covalently bound via the SS bond called the J chain. So this is the J joining chain, J for the joining chain that hold the FC region of this 5 antibodies. This chain functions in polymerization of the molecule into a pentamer, present also as monomer on the membrane of the mature B cell. The properties of the IgM is the third most common in the serum, which constitute 5 to 10% of the total serum immunoglobulin. IgM is the first immunoglobulin to be made by the fetus and the first immunoglobulin to be made by the primary immune response. As a consequence of its pentameric structure, IgM is a good complement fixing immunoglobulin because it consists from the 5 antibodies, so it is more potent in complement fixation. IgM is also a good heme agglutination antibody. So in the blood group, so the agglutination of the RBC is due to IgM. IgM bind to some cells via Fc receptors. The another immunoglobulin is the IgE, which exists as monomer, this is the standard antibody, and has an extra domain, the constant region, had 4 CH domain. Molecular weight 190,000 Dalton. Property: it is the least common serum concentration, which is constituted about 0.002% of the total serum immunoglobulin. Since it binds very tightly to the Fc receptor on basophil and mast cell even before interacting with the antigen. So it is present in low concentration in the serum because it is bind to the cells, basophil and mast cell. It is not present free in the serum. Involved in allergic reaction as a consequence of its binding to basophil, mast cell, binding of the allergen to the IgE on the cells result in the release of various pharmacological mediators that result in allergic symptoms. It's also called homocytotropic antibody, meaning that bind to cells, and called reagenic antibody. Homo is like cells because it has affinity to bind to cells through binding to the Fc receptors. IgE plays a role in the parasitic elements and the protozoal disease. Eosinophils have Fc receptor for the IgE and binding of eosinophil to IgE-coated elements, resulting in killing of the parasite. IgE does not fix the complement. So it is in the allergy, it's bind to the mast cell and basophil. This is in the red one, which is important. While in the parasitic and elements and protozoal disease, it's bind to the eosinophil because the eosinophil had Fc receptor for the IgE. IgD, the last one, which is this structure, exists as monomer and molecular weight 150,000 Dalton. Properties: IgD is found in low levels in the serum, constitute about 0.2% of the total serum immunoglobulin. Its role in the serum uncertain. IgD is primarily found on the B cell surface, on the cell surface of the B cell, where it functions as a receptor for the antigen. IgD does not bind to the complement. Variation of the immunoglobulins. We discussed previously the isotypes, which is all the classes and subclasses of the immunoglobulin that are present in normal individuals: IgG, IgM, IgA, IgE, IgD. While allotype, there is a single amino acid difference in the peptide chain, in the constant heavy chain and constant light chain, so it is called allotype. The idiotype, the unique amino acid difference in the sequence of the variable heavy chain and variable light chain. So allotype in the constant region, while idiotype in the variable region. And thank you.